Hey guys, my name is Shelton and I'm a junior student at Victoria University studying computer science and applied mathematics. My research is multi-objective evolutionary clustering of single-cell RNA sequence data. As we all know that cells are the main building block of human bodies, and scientists are very interested in studying the heterogeneity of cells from different organs and tissues, and thanks to the single-cell RNA sequence technology, after 2009, scientists could now review the full transcriptome of cells in cellular level. Such invention marks the end of Human Genome Project and the start of human cell analysis. By cataloging cell types and cell states, scientists could now develop new drugs, regenerate new medicine, and more importantly, diagnose, treat, and monitor human disease and disorders. Our research aims to catalog cell types by clustering single-cell RNA sequence data. The main idea is to divide individual cells into groups such that the gene expression of cells within each group is similar to each other and dissimilar to the expression of cells in other groups. Here are several challenges of clustering scRNA-seq data because of the complexity of single-cell RNA sequence technology. And the first is the technical challenge. We have a high technical variations with over 50% of dropouts, which correspond to the zero inflated value in our data sets. And then we have our computational challenge. We have an average of 2,000 instances with 20,000 average dimensionalities. In order to solve this challenge, we have our data pre-processing pipeline. We have our count matrix, and then we did filtering. We transform the data, we select tally variable genes, we did scaling, and then we reduce the dimensionality. Another challenge is that the goodness of clustering cannot be formally defined. So here, clustering is actually converted to an optimization problem with two objectives of separation and cohesion of clustering. In reality, most approaches use single objective evolutionary algorithm, which might not review some meaningful implication from the dataset and map to the real-world application. So here in our research, we use multi-objective evolutionary algorithm. Here's a general architecture of the algorithm. And in our study, we design both multi-objective evolutionary algorithm and single-objective evolutionary algorithm. MOEA was compared with SOEA with baseline algorithms, k-means, and phenograph. We tuned the hyperparameter of MOEA with 7 standard machine learning benchmarks from Q repository, and then we tested the performance, stability, consistency, and efficiency of MOEA with 48 single-cell RNA sequence reference datasets from IC bench package. And then we tested the accuracy of the algorithm with 6 synthetic datasets with true labels and known number of clusters. And then all experiments were executed 30 times in high-performance computing resources. We have our internal validation matrix, COA coefficient, and external validation matrix, normal mutual information, adjusted random index. The first experiment is the internal validation. The results show that the performance of MOEA and SOEA is on par with the performance of baseline algorithms, and MOEA can outperform baseline algorithms in some datasets. The second experiment is the external validation. The results show that the accuracy of MOEA and SOEA is on par with baseline algorithms. MOEA and SOEA have a consistent performance. The third experiment is the metamorphic testing to test the stability of clustering algorithm. We designed 6 separate metamorphic relationships which are independent to each other. They are basically perturbations to original data sets. We hypothesis that all clustering algorithms are stable after each perturbation as pre-processing step removes the perturbation effect. And by statistical testing, we concluded that all clustering algorithms are stable to minor changes of data after pre-processing. The last experiment is the running time analysis. Overall, MOEA ran longer than SOEA and baseline algorithms. The first graph and second graph, the efficiency of MOEA was improved by 91% when using 32 central computing units with 4 tasks per node and 8 CPUs per task, which is the last graph. Our results demonstrated that the performance and accuracy of the evolutionary algorithms were consistent, stable, and on par with or better than baseline algorithms. Running time analysis of multiprocessing and high-performance computing resources showed that evolutionary algorithms can efficiently handle large data sets. This is my acknowledgement, and thank you for listening.